Great. So we are recording now. We have finished chapters 1, 2, 3. So I'm on to chapter 4 now, and your first assessment, which is a quiz on Monday, will cover those sections in chapters 1, 2, and 3, and what I'm talking about today, which is the beginning of chapter 4. Chapter 4 is all about tissues. The whole chapter is titled Tissues. So I'm going to pull up a whiteboard here and say some very introductory things about tissues and then get on with what you need to know for the quiz. Tissues, the definition of a tissue is just a group of cells working together to perform a specific function. So I just think of it as a group of cells and they all are geared toward a specific function. That's the textbook definition of a tissue. There are 4 major types of tissues in the human body. We're going to detail all 4 of these, some more than others, and I'll tell you why in this section. The 4 major types are epithelial, connective, muscular, and nervous. So all 4 of these tissues we're going to study in this course. These 2 types of tissues we will study in great detail right now, meaning today and next week. 3 and 4, we're just going to lightly touch those at this juncture, because we will do the entire muscular system later in this course and the nervous system at the end of this course. So we just briefly talk about these 2 toward the end of our tissue section. We detail these 2 in great depth. All tissues in the human body, every single tissue that you have in your body, can be classified in one of these 4 categories. So that's first and foremost to know. Now I'm going to get right onto it with this one. We're going to talk about number 1 today in great depth. So I'm going to just call up a new whiteboard. I can't talk and write about every single, sorry about the writing. It's a challenge, as you know, this is a glass surface that I'm writing on with a plastic pen. And then I have to click 2 different buttons to get an eraser out. All right, so we're at epithelial tissues. When I look at this word, and what I recommend you do is you just think of a tissue that covers or lines a surface. That's what these do. So obviously, the easiest thing to think of is your skin, that the outer layers of your skin are epithelial tissue, but also things that line the inside of our organs are epithelial tissues as well. For example, cells that are tissues that line the stomach and your intestines and the air spaces in your lungs, yada, yada, yada. So it's not just something that covers the outside of a surface, but also lines the inside of a surface. The lining of our blood vessels are epithelial tissue. So now I'm just going to give general characteristics of all epithelial tissues, and then we'll get into specific types. The first one is polarity. These are straight from your book, but I'm going to just briefly explain what these words mean. But polarity just means that the environment on one side of the tissue is different from the environment on the other. That's what polarity refers to. And so we say that epithelial tissues have an apical surface that usually faces the outside of the tissue, and a basal surface that's what anchors the epithelial tissue to deeper tissues. So those are this is exposed to the outside air somehow, to the environment, and this one is attached to deeper tissues. So the environments are different from on this side and this side. That's what the word polarity means. Second characteristic. Your book calls this characteristic special contacts. All that means is that epithelial tissues have close fitting cells. They're tightly knitted together. That's what special contacts means. And they're knitted together through specific cellular connections called tight junctions and desmosomes that create that special contact. Third, all epithelial tissues are supported, that's a U, supported by connective tissue. That's on the basal surface, obviously. So usually there's a reticular type of tissue and some sort of a basement membrane or basal lamina of connective tissue that just helps glue the epithelial cells or epithelial tissues to deeper tissue. This is a big one, so I'm going to put a star next to it. All epithelial tissues are avascular. That means they do not have a direct blood supply to the cells. So epithelial tissues don't have their own blood supply. They get their nutrition through diffusion from deeper tissues. Finally, epithelial tissues have a high ability for regeneration. We replace either dead or damaged epithelial tissues very easily because there are stem cells located in epithelial tissue surfaces so that they are constantly, I'm just going to write, almost constant cellular division. Stem cells provide, let me just mute, mitosis constantly, which is just a form of cellular replication. So we have stem cells usually where we find epithelial cells, not always. They have a high propensity for regeneration if they're lost or damaged. Let's go to a new whiteboard. Now I'm going to talk about specific epithelial tissue classification. The last board was about characteristics. This is how we classify different epithelial tissues. And we classify them based on shape of cells and number of layers. All specific epithelial tissues are classified based on the shape of the cell and the number of layers in that tissue. And so for layers, we have two different choices. We either have one layer or more than one layer. So if the tissue is a single layer, we call that tissue simple. If it has more than one layer, we use the name stratified in that tissue's name. So simple just means a single layer. Stratified means more than one layer of cells in that particular tissue. Now, cell shape classification, we have three choices, not two. And they are, some people say squamous. I was taught to say squamous. It doesn't matter how you say it. So squamous just means flat cells. Cuboidal means cube-shaped cells or box-shaped cells. And columnar means tall cells, column shapes. The word column is in columnar. So all epithelial tissues are going to have one of these two names one of these two words in its name, and one of these three words in its name. There are some exceptions to this, and we'll talk about them when we get there. So let's just, I'm going to call up a new whiteboard, and we're going to start classifying or identifying individual epithelial tissues based on this classification system that we just outlined. And before we go any further, I'm going to tell you that I recently posted a study guide, a tissue study guide on our Module 1 for you to print out and use. What that is a study guide of all the tissues, the specific tissues in their categories in the human body that we are going to study, okay? And so best idea if I were you is I would just call up that, it's under a file, so it's a PDF file. You can just click on it, it will pop up. You can print it out on a printer, and then you can just write notes on it about those specific tissues, which is what I would do if I were studying for this quiz. So now we're going to talk about specific epithelial tissues, and we start with the simple tissues, the single layer tissues. So let's do that. I'm just going to write simple. Let's just number these. Simple. I say squamous. Some people say squamous. Epithelium. The name now tells us everything about this tissue. It's a single layer of flat cells that covers or lines a surface. There you go. Now you know what it is. What I'm most concerned about with these individual tissues is that you learn a couple locations and a couple functions. So that's what I'm going to mostly go through. These cells, this tissue sort of looks like a tiled floor. Now the cells are not perfectly square shaped. They're shaped like this, but it's a flat layer. If you had a bird's eye view of the tissue, it would look like this. If you had a side view of the tissue, all you would see is these flat cells knitted together like this. You might see a nucleus in each cell. So that's kind of what it looks like, simple squamous epithelium. Now we're just going to do functions, and then I'll do locations after that. Two main functions of this tissue. Diffusion and filtration. These are almost the same thing. What both of these words basically refer to is the movement of something from this side of the tissue to this side. Because simple squamous epithelium is so thin, it's so paper thin, it's actually even thinner than paper, things easily move from one side of this tissue to the other through the process of diffusion or filtration. Don't worry about the difference between these two words right now. They both mean movement across the membrane. Diffusion is a passive process. Filtration is also passive. It doesn't require energy, but it usually requires some sort of pressure. Now, locations. There are more locations in the human body than I can possibly list right here. So I'm just going to give you two very common locations. Lung alveoli, that refers to the little air sacs in our lungs, deep down in our lungs. And kidney glomeruli, that's plural. These are the filtration sites within our kidneys where our kidneys filter urine. So this is deep in our lungs where gas exchange occurs, diffusion. These are structures that are deep in our kidneys where we push things out of the blood and into tubules to make urine. So that's why the functions are listed as diffusion and filtration. This would be diffusion, this would be filtration examples. Another example of simple squamous epithelium, before I move on to the next specific example, endothelium. If you see this word, or you hear me say it, or you read this word, it's basically the same thing as epithelium, only it specifically refers to something that lines a surface. So it's thin, simple layer of squamous cells that line a surface. Example would be, I'm going to write simple squamous endothelium lining blood vessels. Okay. All right, let's move on. So, number 2 on our list is going to be simple cuboidal epithelium. These are all in the study guide, by the way, that I posted on the Canvas page. I'm going to abbreviate epithelium just so we get out of here a little faster. You know what this means now? It means a single layer of cube-shaped cells that covers or lines a surface. So they're box-shaped or cube-shaped cells, so they sort of have a three-dimensional. This is an exaggeration. They're not perfect squares, but they kind of look like this. And they would be sort of lined up next to each other. Tissue that looks like this. So it's a single layer. Their functions are slightly different than diffusion and filtration. We say the functions of this tissue are secretion. That's an R, S-E-C-R-E-T-I-O-N, and absorption. Oftentimes, we find this type of tissue in glands that secrete things, like sweat glands, digestive glands. Absorption means moving across, just like diffusion or filtration. Absorption means moving something through this tissue to the other side. And classic locations, just to give you 2 of this tissue, would be a different place in the kidneys, kidney tubules, and those perform a process called reabsorption. And a second classic location would be gland ducts. I'm going to write that has glandular cells have secretory functions. So I'm going to write secretion. And let's move on to number 3. Simple columnar. Epithelium. The name tells us what it is, a single layer of tall column-shaped cells that covers or lines a surface. I'm just going to give you classic locations. The functions of this is absorption. That means moving across the surface. A-B-S-O-R-P-T-I-O-N. And secretion as well. So these cells can have secretory capabilities and absorption, just like these up here. The only reason that we switch these 2 functions in order is because most simple columnar cells do this more than they do this, and most cuboidal cells do this more than they do that one, but they perform both. Locations, classic locations in the human body of this tissue is pretty much anywhere along the digestive tract. So you could pick, I'm going to write that first. You could pick stomach, any of the intestines, all the way down to the end of the digestive system. The last section of the large intestine is called the rectum. All of these have secretory and absorptive capability. And number 4, I'm running a little bit out of space here, but it's our last epithelial. This is a special exception to the classification rule, and it's got a long name. Pseudostratified columnar epithelium. This pseudo, the word pseudo means false or fake. So it's a fake stratified appearance of tall column-shaped cells. So technically, this type of tissue is simple columnar epithelium, but typical simple columnar epithelial cells look like this under a microscope. They're tall column-shaped cells that are all lined up next to each other, and they have some sort of a connective tissue holding them down. And we can see the nuclei in each one of these cells. Pseudostratified columnar epithelium, here's the basement membrane, the connective tissue holding the cells down. It looks like this. It's not as nice and neat. It looks like it's more than one layer of cells, but technically it's not. Each of these cells has a part of the cell that touches this connective tissue. So even though it looks like it's more than one layer, it's really just a jumbled scenario of simple columnar. So pseudostratified just means a false stratified layer of columnar epithelial cells. Usually this tissue, the cells are ciliated. That means they have little hair-like extensions coming out of the cells like this. Functions of this tissue are largely secretion. They secrete mucus and movement. That means movement of debris along their surface. And you'll see why when I give you the classic location. The classic location of this is the upper respiratory system. So the trachea and associated structures are the biggest. We have this type of tissue lining our airways, our major bronchi and the trachea, and they secrete mucus. That's why we clear our throats multiple times a day just from mucus secreting from these cells. Also, we inhale things, dust and debris, into our airways all the time, and these little cilia help propel that stuff back up out of our airway. So hence the function, secretion, and movement of debris. All right, that takes care of all the simples. Now let's do the stratifieds. I'm not trying to blast through here super fast, but I know that it's Friday, and most likely you want to get through this just like I do. So of the stratifieds, I'm just going to label them 1, 2, and 3. Stratified squamous, epithelium. That means more than one layer of flat cells that covers or lines a surface. The stratified functions, always, when you see this tissue anywhere in the human body, from now on, think protection. That's its biggest function, its main function. And the reason is because we find this tissue at entrances and exits of the human body. So any area of abrasion, we find this. So I'm going to give you, okay, there's more than this list is much longer than I'm going to give you. I'm just going to give you some examples. Skin. We have this tissue lining the mouth and the upper esophagus. The other end of that digestive system, the anus, the female vagina. All of these areas of the human body are exposed to the exterior world, and so we need protection at these junctures. I'm just going to write esophagus. That's the tube that leads from the mouth to the stomach. All right, let's move on to number 2 of the stratifieds. And we're going to combine stratified, cuboidal, and columnar into one category. The reason is because they're both quite rare. We know from the names what they are: more than one layer of cubed or column-shaped cells that cover or line a surface. First thing I'm going to say is these are very rare. We don't find them in a lot of places in the human body. Locations typically in the ducts of large glands. And in parentheses, I'm just going to write the male reproductive system, which you will learn all of those structures, the glands, when you take A&P2. We also find these in mammary glands. And also in some types of sweat glands as well. Not all sweat glands are stratified cuboidal, but some of them are. These have a dual function of secretion, and because they're stratified layers, some protection as well. And finally, the last stratified, I need to just call up a new whiteboard layer over this one. So we write number 3. This is a special exception. And it's called transitional epithelium. Its name doesn't really tell us that much until you learn what it is and what it does. This is a special type of stratified tissue that is only found in the urinary system. So locations, specific locations, would be the lining of the urinary bladder, also the urethra, which leads from the bladder to the exterior world, as well as ureters, the tubes that run from the kidneys to the urinary bladder are lined with this type of tissue. It's a special tissue. A picture speaks 1,000 words. So when, for example, if the urinary bladder is just a holding chamber, you know that for formed urine. When our urinary bladder is empty, the cells that line the interior of the bladder. So actually the urinary bladder, if I draw it, the inside looks like this. It's not a smooth, perfectly smooth surface. It's variegated. If we magnify cells along this lining, they look like this. So this would be the lumen, or the inside of the urinary bladder. This is all deep connective tissue. And the cells that line are these plump cells that sort of pile up on top of each other. That's why when we talk about this tissue, we talk about it with the stratified layers, because when it's relaxed, when the bladder is relaxed, the cells sort of pile up on top of each other. If we fill this chamber with fluid, it expands And these cells have an ability to sort of stretch past each other. And what happens is they stretch out and become more of a simple type of tissue like this. The ability for these cells to slide past each other when the contents fills up, when the lumen fills up with fluid, gives its ability to stretch. So function, the main function of this tissue is a word called distensibility, distension, which just means the ability to stretch. Now, before we end this lecture, I'm going to quickly talk about glandular epithelia, and we're going to just classify a few different types of glands as your book does, and then we'll be done for the day. And I'll take any questions you have. So I'm going to actually call up a whole new board, so I have to erase. If you have any questions about these old boards, just let me know at the end. So I'm going to call up a brand new set of boards and just write glandular epithelia. Obviously, we're just talking about glands now in the human body. The definition of a gland, and I'm making air quotes right now, is just one or more cells that synthesize and secrete a product. So these cells actually make some sort of secretion and then they release it. So that's the definition of a gland. The secretion that comes from these cells can be water-based, they can be lipid-based, fat-based, things like steroids. We classify glands basically on these types of criteria. So I'm going to put based on typically location of secretion, and that would be using the words like, I'll explain this in a minute, exocrine or endocrine. That's an N, E-N-D-O-C-R-I-N-E. So the first classification criteria is location of secretion. Usually the other criteria is number of cells. And we call the gland either unicellular if it has one cell, or multicellular. Now, I'm going to talk about these two terms really quickly. Endocrine, you've heard that word before, endocrine system, endocrine glands. Endocrine glands are defined as ductless glands. A duct, D-U-C-T, is just a tube. That word just means a tube. So these don't have a tube. They're ductless glands. They always secrete hormones. Hormones are chemical messengers that are carried by the blood to their target cells. So secrete hormones that enter the bloodstream. The word exocrine, that refers to glands that have a duct that means a tube that lead to or secrete their contents into a specific site, like a specific organ or a body surface. So really quickly, endocrine glands are just a group of cells that synthesize, make, and secrete hormones, these little chemical messengers that enter. They don't have tubes or ducts. The secretions, the hormones, enter the bloodstream. So there's usually a blood vessel, a capillary, very close to an endocrine gland. And the hormones that are secreted by these cells just enter the bloodstream directly and then use the blood to go everywhere in the human body until they find their target cell. Exocrine glands are typically a group of cells, almost always. I'm running out of space, so I need to redraw my picture here. My gland is going to go this way. It's a group of secretory cells like this that have a duct or a tube that's lined with cells, cuboidal cells. And the secretions, usually a fluid, not a chemical messenger, but a fluid that comes from these cells, enters this tube and goes directly to some sort of an organ or body surface. Examples of exocrine glands would be things like sweat glands. So I'm going to write sweat. Mucus glands. Oil glands in our skin, sebaceous glands, salivary glands in our mouth. You can see there's a whole bunch of them. The liver is actually a gland. It's the largest gland in the human body. Last one I'm going to write is pancreas. These are wildly different things, but they're all examples of exocrine glands. Now I'm going to talk about unicellular versus multicellular. That's not difficult at all. Unicellular just means one cell. So I'm just going to give you some examples. That's an N, U-N-I-C-E-L-L-U-L-A-R. So unicellular glands would be things like mucus cells, that we find in our respiratory tract, for example, goblet cells that we find in our GI tract, our digestive system. These are individual cells that secrete something, that secrete mucus. So I'm just going to write example of unicellular. They produce, I'm going to talk about this word really quickly because it's going to come up. If you take AMP1 and AMP2, the difference between this word, mucus, and this word, mucus, this one is the actual stuff, the secretion, that sort of gross green stuff that you cough up when you're sick. That's what M-U-C-U-S, it's a noun. M-U-C-O-U-S is an adjective that describes usually a cell type or gland type that secretes the stuff. So this is an adjective describing a type of gland or cell that secretes this stuff, mucus. All right, so multicellular examples. These are classified, I'm going to write classified by structure. And it's not difficult to understand this. They're either simple or compound. That just refers to how complex they are. For example, a simple gland might look like this. It's got multiple cells in it. This is an example of a simple gland. There's multiple cells that produce a secretion that travels through its duct and ends up on a body surface or in another organ. A compound gland might look like this. I'm just going to draw the same surface. It might have the same main duct leading to the surface, but then it's got several branches like this. That's the only difference. Compound just means it's a more complex structure, but it does the same thing. The secretions are produced by these cells in the gland, and they all lead to a main duct that releases the secretion. Finally, last thing I'm going to say today, I promise, before I take questions, is we'll talk a little bit more about glands when we study the skin, which is coming up next week. Classification of secretion. Merocrine, M-E-R-O-C-R-I-N-E, merocrine glands. The definition of a merocrine gland is that the secretion is by exocytosis, but here's the important part. So I'm just going to write secretes by exocytosis, and the cells, the secretory cells, are not damaged in the secretion or secretory process. They can be reused again. Examples of this would be things like sweat glands, salivary glands. That's opposed to something, a different type of classification by secretion would be a holocrine, H-O-L-O-C-R-I-N-E. These are also glands that release secretions, but the secretory process is usually by the cells rupturing, and they're destroyed in the process. So I'm going to write secretory cells rupture. I'm going to write destroyed in the process. So every time a secretion is made by these cells, the cells themselves, the secretory cells, are destroyed, and we have to make new ones in order to continue the secretion. Classic example of these: sebaceous glands, or what you and I call oil glands of the skin. These, the dead cells, the ruptured cells, are actually released in the secretion itself. All right, guys, we made it to the end. That's good. It was nice and quick. I will post this recording, don't worry, on the Canvas page. It takes a couple hours. It's a long process. I don't want to bore you with the process, but it takes me a couple hours to go through this whole process of posting it on Canvas. So it usually doesn't show up until very late in the day. Sometimes I get to it sooner, a little bit sooner than other days, but definitely by the end of the day, I will post this lecture. Let me just get rid of this board. And if you have any questions, either about the material or about the quiz that's coming up on Monday, let me cut my recording, but you can